Hi everyone, today we're going to cover Unit 3, which is the genital tract infections and cultures. There are some sexually transmitted diseases that I want to bring your attention to, including HIV and AIDS. There's about 18 to 19 people infected for every 100,000 people with AIDS. Other sexually transmitted diseases that we're going to talk about would be chlamydia, genital warts, which is caused by human papillomavirus, gonorrhea, pubic lice, syphilis, trichomoniasis. Those are just a few of the sexually transmitted diseases that we're going to talk about in this lecture. You do need to know the components of the genital tract for males and females in order to understand which types of specimens that we culture. Okay, urethritis is the #1 STD syndrome reported in men, and in females it's cervicitis. So I want to talk about the male internal components of the genital tract and the male external components. So the male internal components would include the testes, the epididymis, seminal vesicles, urethra, and prostate, while the external anatomy would include the penis and the urethra. Internally, for the female anatomy, that would include things like the ovaries, the fallopian tubes, the uterus, the endometrium, the cervix, the vagina, and the urethra. And externally, the components of the female genital tract would include the labia and the urethra. So the most common and appropriate specimens to collect for genital culture in males would be urethral specimens, and in females it would be endocervical swabs. Okay, in the male, the mini tip swab is collected by inserting it 3 to 4 centimeters into the anterior region of the urethra. And in females, there's a catheter that's inserted into the endometrial cavity. There's a mini tip that is a swab that is actually transferred through that catheter and inserted a few millimeters past the cervix. So for males, the appropriate specimen for culture is the urethral specimen, and for females, the appropriate specimen for culture would be endocervical specimens. So we treat urethral specimens a little bit different than we treat endocervical specimens in females. For males, if we grow anything out on the culture, we do want to identify or line list all the organisms identified. We don't want to report it as normal flora because in the urethra of males, there is no normal flora. Any flora that is isolated is either going to be the pathogen or skin flora. Now, pathogens can be determined by the physician, but we do report everything. A proper collection technique should yield only the true pathogen and limit all that skin flora. If the specimen is collected properly, there should be no skin contaminants. Some probable skin contaminants in male urethral cultures would be things like coag-negative staph, diphtheroids, alpha streps, and gamma streps. Now, in endocervical swabs, we often pick up normal vaginal flora, and that flora is going to vary depending on the pH of the vagina. Usually, in the female genital tract, there is an abundant amount of lactobacillus, and lactobacillus is normal vaginal flora that makes lactic acid and keeps the vaginal pH slightly acidic at about 4 to 5. Most pathogens don't like that pH. That flora is going to vary with the amount of vaginal secretions being produced, and also the flora is going to vary depending on their estrogen levels. There are a number of organisms that are completely normal in the vaginal region, including coag-negative staph, micrococcus, a few colonies of enterococcus. If there's a pure culture of enterococcus, that could be a pathogen because enterococcus is a strep of the gut, and that could actually be contaminating flora from the anus. Strep viridans is normal. Peptostreptococcus is an anaerobe, and it's normal. Bacteroides is an anaerobic gram-negative rod, and it's normal vaginal flora. And then other miscellaneous gram-positive rods, including diphtheroids and lactobacillus. Strep B is totally normal in females. However, it can be a pathogen if mom is carrying a baby. It can be a problem for the baby. If you have low numbers of Staphylococcus aureus along with mixed flora, that can be completely normal. And if you have just a few enterics, low numbers of enterics that are contaminating flora from the anus, that can be normal as well, as long as there's a bunch of other normal vaginal flora. So vaginal samples or endocervical samples can be a little bit difficult because you're always trying to ignore the normal flora and be able to deduce what the pathogens are. Now, these genital tract pathogens are, for the most part, considered sexually transmitted, but not all of them. Trichomonas vaginalis is a parasite that can contaminate the genital tract of humans. Candida albicans is a type of yeast. NEISSERIA gonorrhoeae is a sexually transmitted pathogen. Treponema pallidum is what causes syphilis. Haemophilus ducreae can cause the chancroid in people's genital tracts. You can also have group A strep, chlamydia trachomatis, herpes simplex virus, listeria, and strep B. Again, it can colonize mom, but it can become a pathogen if mom is carrying a baby. And then Staphylococcus aureus and Staphylococcus aureus and enterics, rather, if they are there in moderate to high numbers, they can be pathogens as well. Now, one organism that's not on the list but should be is Gardnerella vaginalis. In Gardnerella vaginalis, we're going to talk more about this organism at length, but it can cause what's known as bacterial vaginosis. It's not sexually transmitted. It's actually an organism that lives in the female genital tract, and it can be a problem when it is out of whack with the amount of normal flora that's there. So let's go ahead and start by talking about Trichomonas vaginalis. There are a number of species of Trichomonas, but the one that we're concerned with in this lecture is Trichomonas vaginalis. It is a protozoan parasite, and is usually diagnosed by finding that motile flagellate in a saline wet prep. Okay? Twitching, it has a twitching motility, and you can see that in a fresh wet prep with saline. The motility is apparent when it's first examined and immediately, but it's not going to be motile for very long. A good culture media is not available, but a good culture media is available, but how good it is questionable. Culture is the gold standard, but the media isn't readily available or doesn't work very well. Serologically, we can identify Trichomonas vaginalis with an antigen test like a FIRM or the OSOM Rapid Trichomonas test. These kits are about 80% sensitive, and there is a development of PCR in progress for identifying Trichomonas vaginalis. Let's take a look at a picture of Trichomonas. So here's a drawing of Trichomonas, and you can see that it has 4 anterior flagella and an undulating membrane, and that's what causes the twitching motility, that in the flagella. On the upper right is a gram stain of Trichomonas, and that's very difficult to identify from a gram stain. And then finally on the bottom right is a picture of the Trichomonas in a vaginal wet prep. And if you click on that link should take you to a YouTube video, or copy and paste the link into your browser, and you'll be able to see the YouTube video of motile Trichomonas. Candida species is a yeast that can be confirmed with what's known as the germ tube test. The germ tube test is a confirmatory test for a yeast known as Candida albicans. It is Candida albicans that is the most commonly isolated yeast. And yeast infections are associated with pregnancy, antibiotic use, diabetes, and steroid therapy. Hormonal changes, pregnancy, ovulation, oral contraceptives, and diabetes can all lead to yeast infections. This is not considered an STD. 75% of all females will have a yeast infection in their lifetime. It can be seen on a wet prep or on gram stain, and if we want to culture Candida or any yeast, we can put it on a media known as sabro-dextrose agar. SABS is selective for isolation and cultivation of yeast. So here's a picture of the most common isolated yeast in the genital tract, and that would be Candida albicans. Candida albicans shows what's known as pseudohyphae, and then at the end of those pseudohyphae are these bulbous structures known as terminal chlamydospores. And Candida albicans is the only yeast that produces terminal chlamydospores. You can see that in the germ tube test, which we're going to talk about at a later time. So even though Candida albicans is the most commonly isolated species of yeast, there are other species of yeast that will look a little bit differently. So in the upper left, we have a picture of budding yeast. There are certain types of yeast that only bud. One would be Candida Candida glabrata, and then one can produce just pseudohyphae but no terminal chlamydospores. An example of that might be Candida parapsilosis or tropicalis. There are 700,000 new cases of Neisseria gonorrhea identified every year in the United States. If you recall from earlier lectures, Neisseria gonorrhea does not grow on blood agar, but it does grow on chocolate and modified Thayer-Martin. It's best to culture it on modified Thayer-Martin with the addition of carbon dioxide. It is a fastidious organism that does require 3% to 10% CO2. If you wanted to identify Neisseria gonorrhea, you could do so by setting up the CTA sugars. Neisseria gonorrhea begins with a G, and it is glucose positive, which also begins with a G. It is maltose, sucrose, and lactose negative. You could also identify Neisseria gonorrhea by setting up the Bacticard test. The Bacticard is going to be Pro positive and Glut negative. If you were to gram stain Neisseria gonorrhea, you would find they are gram-negative diplococci, and they may actually be intracellular or found intracellularly on a direct urethral gram stain. Neisseria gonorrhea is catalase positive and oxidase positive. So here is an image of Neisseria gonorrhea intracellularly placed inside of the polymononuclear cells. Okay, you can see that they are kidney bean shaped gram-negative diplococci that are intracellular. 12 million people get syphilis annually every year in the world. There are about 10,000 cases a year in the United States. Now, during World War II, there were approximately 500,000 cases per year in the United States. Now, Treponema pallidum is the spirochete that causes syphilis. It is a larger and tightly coiled spirochete, and it can actually penetrate the skin, much like Shigella and Francisella can do. Types of diagnostic tests would be DFA, VDRL, which stands for Venereal Disease Research Laboratory, RPR, which is Rapid Plasma and Reagent, and TPPA testing, which is Treponema pallidum particle agglutination. It's also good to identify Treponema pallidum initially with a dark field exam by scraping the chancre, and you can actually see the spirochetes in dark field exam. Treponema pallidum is often called the Great Intimidator because symptoms are nonspecific and resemble many other illnesses. Culturing is the gold standard, but it is very expensive and time-consuming. Here in the culture, we set up what's known as roll tubes, and we inoculate the organisms into the roll tubes. The roll tubes contain cottontail rabbit epithelial cells, and what we're looking for is evidence of those cells being infected by cytopathogenic effect, which is the swelling of cells. Okay, in Treponema pallidum, you can have several different stages of this disease. In the first stage, there is a chancre that appears, and that is a sore that's usually on the genitals. In secondary syphilis, you will see a rash on the trunk of the patient and on their hands and feet. And then in the fourth stage of Treponema pallidum, it can go to their central nervous system, and this is called neurosyphilis. Let's take a look at spirochetes on the differential stain in the next slide. So in the top two images, we can see the spirochetes. The one on the left is an example of a gram stain. The one on the right is an example of what Treponema pallidum would look like on a dark field exam. Down at the bottom, we can see keratotic lesions on the feet and chancres on the neck of these patients. Haemophilus ducreyi is not something that's super common in this country. There's only about 30 cases per decade in the U.S. This is much more common in other countries. In the U.S., it is highly associated with the commercial sex industry. This is the cause of a highly contagious sexually transmitted disease, and here Haemophilus ducreyi can form a chancroid or a soft chancre. It's a little bit different than the syphilis chancre. The chancroid of Haemophilus ducreyi has an irregular edge or an irregular margin. It looks more ragged and pliable. Haemophilus ducreyi is a small gram-negative rod that are described as having a school of fish arrangement. It is a very fastidious organism and may be difficult to grow, but you can grow it on chocolate agar with a little CO2. It is slow-growing, small, yellowish to tan. You can push it across the agar, very similar to how you can push Moraxella catarrhalis around the agar. It is catalase negative, oxidase positive, and pretty inert. You can try what's known as a RAP-ID, which is a Remel cassette microtest for serological identification of Haemophilus ducreyi. In this gram stain image, you can see what looks like a school of fish. This is the morphology or the morphological microscopic appearance of Haemophilus ducreyi. There are 900,000 cases per year of chlamydia trachomatis in the United States. It is the most commonly isolated genital pathogen in the U.S. It is considered a parasite or an intracellular bacterium. They kind of go back and forth on what it is, and it can cause many infections, and many infections are asymptomatic. It's also easily transmissible. It is considered a sexually transmitted disease that can be transmitted from person to person or from mom to baby. In females, it can lead to pelvic inflammatory disease. The specimens are obtained from cervical scrapings or mini-tip collections. It can be diagnosed by cell cultures, Giemsa stains, fluorescent antibody staining, or DNA probes. Okay? It won't gram stain because it has a cell wall similar, but it does have a cell wall similar to a gram-negative organism. Here we have a picture of a Giemsa stain in the top left, where the frothy, bubbly objects are the chlamydia. And then on the bottom right, we have a direct fluorescent antibody where the red dots are chlamydia inside of nucleated cells. These are pictures of CPE, or cytopathogenic effect. It's a tissue culture that shows the cells starting to swell. So in image A on the upper left, these are normal rabbit cottontail rabbit cells that are not swollen, and then they are starting to swell in images B and C, and then finally in image D they are very swollen. This indicates that the living cells are infected with a bacterium or a virus. This is called cytopathogenic effect. There are two forms of chlamydia trachomatis in its life cycle. One is called the elementary body and one is called the reticulate body. The elementary body is the infectious form of the organism. It's pretty small at about 300 nanometers. The non-infectious form of the organism is called the reticulate body. Around 8 hours or so after being infected, the reticulate body starts to form, and that's the non-infectious form. Here we can see at the 0 hour, the elementary body attacks to a host cell. The host cell then phagocytizes the elementary body about 1 to 6 hours later. The elementary body matures inside of the phagosome at about 8 hours and becomes a reticulate body. The reticulate body divides by binary fission and condenses back to an elementary body at about 12 to 40 hours. The elementary body is then released about 48 hours later when the cell ruptures. Let's talk about herpes simplex virus. According to the Centers for Disease Control, or the CDC, there are about 20% of all adults that have genital herpes. That's essentially 1 in 4 males and 1 in 6 females have herpes simplex virus. Okay? Members of the herpes viride group would be HSV type 1. That's usually the type that causes cold sores. HSV type 2, which is usually the type that causes genital herpes, and genital herpes can go from mom to baby. We have human cytomegalovirus, CMV, which is a herpes simplex virus. We also have Epstein-Barr virus that causes infectious mononucleosis. That's a herpes viride virus as well. And then we have varicella zoster virus, which causes chickenpox. Now, the varicella zoster virus can later in life lead to shingles. cold sores from herpes virus is always type 1 virus, but genital herpes can be serovirus type 1 or 2. 75% of the time, genital herpes is type 2, HSV type 2. You can use molecular techniques to best identify genital herpes. The most common viral sexually transmitted disease is papillomaviruses. About 6.2 million cases are identified every year in the U.S. The human papillomavirus, or HPV, causes genital warts. There are 60 different types of genetic variations for this virus. Many studies have concluded that colonization with HPV increases the chance of a female getting cervical cancer. Cervical epithelial cells look abnormal or have a coarse clumping chromatin, and this may be a clue that our patient has HPV. Let's take a look at that dense, chunky chromatin and perinuclear halo in the next slide. So cells infected with HPV show a perinuclear halo around the nucleus of the cell and dense chromatin. There is no permanent cure for human papillomavirus, but some treatments would be cryotherapy, laser surgery, or alpha interferon injections. Gardnerella vaginalis is technically a pleomorphic gram-negative rod. However, on a direct gram stain, it does appear to be gram variable. We can usually discover if a patient has Gardnerella vaginalis by finding what's known as clue cells. A clue cell is a squamous epithelial cell covered in tiny pleomorphic bacteria. The squamous cell looks like it's been pressed in sand, and we're going to take a look at some images of that coming up. You can find clue cells in vaginal saline wet preps, or you can find them on the gram stain. Samples that are positive for Gardnerella vaginosus will emit a fish-like odor when combined with potassium hydroxide. Here, a chemical reaction from amines of bacteria and anaerobic bacteria plus KOH indicates a fishy smell, and you can determine the smell by doing what's known as the whiff test. Gardnerella vaginalis is a very small, almost dust-like organism that grows on your blood and chocolate plates at about 24 to 72 hours. It usually takes a good 24 to 48 hours before you even see that dust-like colony. Okay? It's often more noticeable on chocolate agar. You can confirm the organism ID by putting on a selective media known as V-agar. V-agar is selected for Gardnerella vaginalis, and the organism appears to be very, very tiny and beta hemolytic, and you can see that in the image on the right. You can also take that organism and put it on a HBT agar, which is human blood tween. This bug should grow very well on this media, and because they are lipophilic, they actually really like fat. They're fat lovers, and they like the tween, and so they'll be a little bit larger on HBT media. We usually don't report out clue cells on the gram stain. However, this is an example of a squamous epithelial cell that it looks like it's been pressed in sand with gram variable bacteria. This is a pretty good clue that we are dealing with a Gardnerella vaginalis. More likely, clue cells are going to be identified in a saline vaginal wet prep, and they are reported on saline vaginal wet preps. Here in this image, you can see two normal squamous cells on the upper left and at the top of the image, and then we have the bottom two squamous epithelial cells that are completely covered in bacteria. Those are great examples of clue cells, and again, clue cells are associated with Gardnerella vaginalis. Like I mentioned before, Gardnerella vaginalis is not an STD. It is normal vaginal flora, but when it is in greater numbers than the rest of the normal flora, it can cause bacterial vaginosis. This organism is catalase negative and oxidase negative, and its claim to fame is that it's going to hydrolyze starch. It can break down starch into smaller, more simple components. It's beta hemolytic on HBT and V-agar. It does not require the X or V factors, which kind of differentiates it from Haemophilus species. On chocolate agar and blood, it is very dust-like and very, very small, and usually is much larger than Haemophilus, much smaller than Haemophilus. Haemophilus is much larger than Gardnerella vaginalis. The last organism that I want to talk about is Mobiluncus. This is a gram-positive anaerobe closely related to actinomyces, and it can cause vaginosis when it's growing along with Gardnerella vaginalis. It's an anaerobe associated with nonspecific vaginitis. It usually stains gram-variable much like Gardnerella does, but it has more of a hook-like appearance. It is technically more closely related to gram-positive organisms, even though it stains gram-variable. So that's it. That is everything for Unit 3. Today we've talked about genital tract infections and cultures. See you for Unit 4.